to first start by thanking you for the invitation and thanking all the organizers for this conference. And today I'll be talking a little bit about how we use an RNA-like polymer to address some issues with RNA non-enzymatic replication. And so I'll just start with acknowledgements. All this work was done in Professor Shostak's lab at Harvard. And this was done as a joint effort with Dr. Lijun Zhu. So just bear that in mind that this was a three-pronged effort. And so today I'll be talking about, I'm having a hard time with moving the slides. Okay, so today I'll be talking about two stories, and it's using these 3' dideoxynucleotides as shown here. Maybe I can just put my pointer shown here. And so the first story will be the polymerization pathway, and this is to make longer oligonucleotides. The second one is using these RNA fragments that have terminated with these specific chemical modifications. And we want to see if we can extrapolate or at least generate functions or sequences that would have some type of catalytic function. So obviously, this introduction might be a little bit basic for this audience, but I did want to kind of juxtapose where we're talking about. So really, we're at the interface of the pre-RNA world and the RNA world. And so if you think back to that era or that time, we're really talking about these heteropolymers that can go either through an RNA-like pathway to converge to RNA, or it could be something completely different. And today's talk is on these RNA-like. So we often get the question, why RNA? Well, this has been introduced, I think, over and over again in this session, but the core of the ribosome is RNA. There's many relics, namely these cofactors that have an RNA building block or unit dangling from them. So we do have evidence, right, in modern biology that points towards that RNA. And so when we look at modern biology, we use typically these nucleoside triphosphates. We use these very sophisticated polymerases, and we can replicate the genome, whatever nucleic acid sequence, with pinpoint accuracy and quite fast, right? But going back on early Earth, we wouldn't have these sophisticated catalysts. And really, when you look at these nucleoside triphosphates, they're really not that reactive. So Orgel, a pioneer in this field, came up with these phosphoroimidazoles, and this restores some of this chemical reactivity that would be used for genome replication on early Earth. And so you'll have to take my word for it, but if you can imagine that the first template was already present, we envisioned these mononucleotides binding on and off this template. This triggers polymerization events leading to full-length products. And then the second step would be denaturing these two oligomers that would allow for a second round. Now, despite more than 50 years of research on this type of scheme, we still have large hurdles to overcome before we can demonstrate or successfully demonstrate one full cycle of replication. And so one difficulty that I wanted to address was the hard times that we have with copying all 4 nucleotides, and particularly AU-rich sequences. This seems to be an issue. Also, the gold standard in literature at the moment is that we can copy up to 7 nucleotides quite well in RNA system, but this is simply not efficient enough to generate sequences that would encode for function. And so when I started in the Shostak lab as a bioorganic chemist, I thought, okay, well, we can improve this in one of 3 ways, right? We can play around with the leaving group, but this had already been attempted by a talented postdoc in Jack's lab. You could play around with the nucleophile, so make that hydroxyl more nucleophilic, or you can play around with the scaffolding to enhance the binding. And these attributes would enhance, right, that primer extension. And so we use these model experiments called primer extension experiments. You have a shorter strand, a templated region, you throw in these activated mononucleotides, and you want to see how well this extension goes. And just to remind you, these are the 2 problems that I'm trying to address. So we're really thinking about alternate genetic polymers. And although there's been many different chemical modifications that have been surveyed, in various groups. The one I'm going to talk about today is called the NP system. And so this is a chemical mimic to DNA, right? It resembles DNA because you're just swapping out this amino group for this hydroxyl. But in reality, this is a structural mimic to RNA. So it adopts this 3' endo sugar pucker. And so when it's in a polymeric strand, or even as the free nucleotide, it resembles a lot better RNA than DNA. And so I wasn't the first to work on this. There were two papers that had come out of Jack's lab, mainly on the kind of leaving group version 2.0, and this was now leaving group version 3.0. And so the question I wanted to tackle was, with the advent of this new leaving group, would it act as a synergistic effect with a more nucleophilic group as the nucleophile? And so I went in the lab and made all sorts of nucleotides, some that were resembling what modern biology has, and maybe other more exotic ones. And I'll just be talking to the ones that are highlighted in red here. And so as an organic chemist, I joined the lab, and I didn't really enjoy how these mononucleotides, these activated mononucleotides, were prepared. So most pathways were using HPLC purification, and so it wasn't really scalable. So I first tackled this, and so on this slide, although it seems quite simplistic, this was actually a lot of work. So you can obtain these commercially available building blocks. You could protect this exocyclic amine with an FMOC protecting group, and do some Yoshikawa type chemistry to install that first monophosphate. And then it's a matter of finding conditions to activate this with the imidazole, and conditions to remove this FMOC without hindering or degrading this portion of the molecule. And so I developed a scalable and generalizable pathway for all these 4 nucleotides, which was great. You could do this on now 500 milligrams or even the gram scale. And so the first set of experiments was to see, can we copy homopolymers? So polymers that have, either stretch of C's, G's, or A's and T's. And we were anticipating pretty good results with G's and C's. And sure enough, this is what we see from this page experiment. Right after 10 minutes, you get full extension. So we were not surprised and quite content with this. But we could also see a decent extension with A and T, albeit a lot slower. So this is on the order of days. And if you leave this reaction actually further or longer, you do converge to the full length product. So we were satisfied with these results. We knew that we could copy efficiently with these types of monomers. But that test was more, can we copy all 4 nucleotides in one pot? And sure enough, this can be done quite easily. And you can see here from just within a day, you do extend to the full length product. And so we were quite happy with this, and at the same time, a little bit surprised, because it turns out with the RNA system, you're kind of blocked at the 4 position, and you have to use some auxiliaries. So this has no auxiliaries. This could be done at high salt, at low salt. You'll get the same kind of full extended product. So that's great that we're extending, but are we doing this in high fidelity or not? And so what we did is just general mass spectrometry on this band here. And what we were able to show is that it is the primer plus the right composition. Now, this doesn't give you an idea of fidelity per se, because the sequence could be scrambled, but we did have evidence that if you do hit a mismatch, the activity of further or subsequent extension is much slower. So we anticipated that, yeah, this is probably high fidelity process. And so no other species was detected except our desired composition. So we had indication that this was a high fidelity process. But we wanted to reach that kind of 30 nucleotide mark, right? This is where sequences get long enough to encode for useful function. And we had the example of a hammerhead ribozyme, right? And so we did this in a stepwise fashion. We could copy 12 nucleotides, no problem. We can get this up to 18 nucleotides again within just a few days. But I guess the more important part, and this was done with an average single nucleotide extension that was comparable to RNA, but for much longer stretches. the important tidbit was that we could now extend all the way up to 25 nucleotides. And this was great because if you add on the primer sequence length, then you exceed that 30 nucleotide mark. And this is where we concluded this study. And so the next logical question that we asked was, okay, if this works really well, and this was obviously a striking step from the 5 to 7 nucleotides that we could copy in the RNA system. And so if we can polymerize this really well, what about ligation? What can we do with that? And ligation does offer multiple advantages, right? Fewer reactions to get longer sequences. Presumably it could copy through secondary structures. It should have no problem copying AU-rich sequences. Drawbacks include that they're not prebiotically plausible without the presence of monomers, obviously. And you could encounter the error catastrophe, the inheritance catastrophe, because it should accommodate a lot more mutations. But nonetheless, it turns out that ligation experiments were quite scarce in the literature. So Legion had characterized a lot of this ligation pathway in the RNA system and compared it to the NP system. So here, the only difference is that the terminal nucleotide has this 3' amino. And so what we showed was that you can enhance the ligation rate up to 150-fold when you just replace this ribose with this 3' amino system, which was great. We also showed that you could copy mixed sequences, no problem. So these are just 4 tetramers adding, within 20 hours. So this was great. If you leave it for longer, it does converge to the full-length product. But now that we know that ligation works well, what can we do with it? And so we wanted to demonstrate, and we looked in the literature. There was no, to the extent of our knowledge, clear transition from a pre-RNA world to a full RNA world. And so the idea here is that you would take these short fragments and you would assemble them into something that would have some type of beneficial function. The crux of the problem was that RNA synthesis or RNA ligation is too poor to generate anything of this magnitude. And if you look at just a 3' NP-DNA system, there's no sequences out in the literature that has enzymatic activity. So we were kind of at a standstill. And so the catharsis that we kind of reached was whether we could use a chimeric genetic polymer. So, an RNA, almost fully RNA, but where we need to ligate, we use the power of this more nucleophilic system. And so the concept is shown here. We would have self-replicating components, small short oligonucleotide strands. These would base pair in a splint-directed fashion. So the green sequences are just splints. The blue ones would be active. You would generate something non-enzymatically in a function that would encode, a sequence that would encode function. The splints would have to be removed somehow. And then this newly formed ribozyme, and in our case, we're using a ligase, could exert its function. And in this case, it was the self-replicating ligase that Joyce had come up with in 2014. And so this is the ligase. These are the different fragments that we joined together. Most of it, as you can see, is RNA, but at the ligation sites, these are the 3' amino nucleotides. So they're kind of sprinkled in the scaffold. And so the first time we assembled this was using these RNA splints. And we were really excited because on the left here, you can see that ligation experiment gave us our 52 nucleotide product within a day. So super excited about that. But when we threw in the substrate, increased the temperature to hopefully remove these splints, we saw no activity. So that was a little bit of a bummer. And so we attributed that to strand inhibition pathway. And so Legion and I worked on developing new and kind of innovative ways to disrupt this type of strong binding. And I'll just show one example here where we swapped out the RNA fragments to DNA splints now. And here we can restore activity quite well. So you can see on the left hand, the non-enzymatic process works well. When you add the substrate, increase the temperature, you get very nice ligation within 2 hours. And so I'm getting to limit here. I hope I've convinced you that this 3' RNA system has vast potential. On the polymerization side, we could get products up to 25 nucleotides extension. on the ligation, we could produce potentially sequences that have enzymatic activity. And without maybe doing this on purpose, we did show that mixed polymers are compatible with sequences that encode function. So this is a little bit of a tantalizing idea, because on prebiotic Earth, many have shown that multiple different genetic polymers could have been present. And so with that, I'd like to thank my collaborator, Lejeune, and my advisor, Jack Shostak, and all these funding agencies, and you for your time. Happy to answer questions.